Hi, everyone. Greetings from Spain. So today I'm going to be telling you a story about adenoviruses. As probably many of you know, adenoviruses are best known as therapeutic vectors, both in experimental setups and also in real life setups. Like, for example, many of the SARS-2 coronavirus vaccines nowadays in use throughout the world are based on adenovirus vectors. Adenoviruses are also serious pathogens in humans. They can be quite serious in immunosuppressed individuals. And also in animals, there are adenovirus pathogens that can cause economic losses, in particular in farm animals. Adenoviruses have been found in all sorts of vertebrates, from fish to humans, but for obvious reasons, those most studied are the human adenoviruses, in particular adenoviruses in species C, which contains human adenovirus type 5. The capsid architecture of human adenovirus type 5 was solved by cryo-EM in 2010 by Hong Xiao and later refined in 2017. It was one of the first high-resolution structures solved at the beginning of the resolution revolution. And the virion is a non-enveloped icosahedral particle, rather large, 95 nanometers from vertex to vertex. Pseudo-T equals 25 icosahedral symmetry. The capsid is composed by the major code protein, hexon, a double jelly roll protein. At the vertices, we have the pentameric penton base and the trimeric fiber, and these are in charge of binding to the receptor. Human adenovirus has a series of minor code proteins on the inner capsid surface, and on the outer capsid surface, it has another minor code protein called protein 9. This protein is involved in stabilization of the capsid, and it forms a complicated structure, this sort of hairnet weaving around the hexons and holding the structure together. You can see here in more detail the structure of protein 9. As you can see, each monomer of 9 starts here at this so-called Triskelion, and then it goes on through a disordered region weaving around the hexons, and it ends in an alpha helix that forms a helical cluster with other copies of protein 9 coming from different triskelions, and some of them even coming from different facets in the icosahedron. Inside the capsid, we have what we call the core. The core contains the viral genome, double-stranded DNA, 35 kilobase pairs long in the human adenoviruses, and a large amount of viral encoded protein. These are the so-called core proteins, which are thought to be DNA condensing proteins, similar to histones in the cell. But several works have recently shown that these core proteins are not just subway pushers helping to squeeze the DNA inside the capsid, but they actually play active roles regulating different aspects of adenovirus maturation and coating. Inside the capsid, we also have some enzymes involved in genome replication, packaging, and maturation of the particle. So there are many studies about structure and assembly of human adenoviruses, but human adenovirus type 5 in particular. But what about the other adenoviruses? We decided to try to expand our knowledge of the structural variability in the Adenoviridae family. And regarding the structures known in adenoviruses, as I said, we have the adenovirus 5 known since 2010. More recently, the structure of an adenovirus with ocular tropism, as opposed to the respiratory tropism of F5, has been solved. Also, there are high-resolution structures of incomplete particles of a bovine adenovirus and low-resolution structures of some non-human adenoviruses. We have lately determined the high-resolution structures of human adenoviruses with enteric tropism and avian adenoviruses. And if you want to know more about these, I invite you to visit Marta Perez-Illana's breakout room tomorrow. But today I'm going to be talking about reptilian adenoviruses. We have determined the structure of adenoviruses infecting snakes and lizards, and these belong to the adenovirus genus. One interesting feature of these adenoviruses is that their capsid is more thermostable than the adenovirus 5 capsid. As you can see here, if we hit adeno 5 at about 50 degrees, the infectivity is completely gone. And in negative staining, we can see that the capsid is completely disrupted. However, the adenoviruses still retain a fair amount of infectivity after hitting at 50 degrees, and we can still find some seemingly intact particles. What about the genome? It was known for several years already that adenoviruses lack some of the structural proteins in the human adenovirus virion. They do not have the core protein 5 or the external capsid, minor capsid protein 9, this protein that forms the hairnet that I showed you a minute ago. Instead, these adenovirus virions contain some proteins that could substitute for the role of 9 and 5, such as LH2, LH3, and P32K. And this was known already for the ovine adenovirus, and we've shown it also by mass spec for the reptilian adenoviruses. Several years ago, we obtained a cryo-EM map at low resolution, 11 angstroms, of a snake adenovirus. And we could see that this capsid has these prominent knobs at the icosahedral threefold and local threefold axis that we knew corresponded to protein LH3 because it had already been shown for the ovine adenovirus. We had low resolution then, but we collaborated with Mark van Rijk, who is a crystallographer, and Tan Nguyen in Mark's group managed to obtain the crystal structure of recombinant LH3. And this is how LH3 looks like. You can see this gold structure here intercalated between the hexon. These are homology models for hexon in gray. So those of you who work on phages in the audience probably have immediately recognized this structure because this is a trimeric beta helix fold, very typical of bacteriophage tail spikes. So at this point, we could say that we have a phage tail spike displayed on the surface of a vertebrate virus capsid. And we can think then of adenoviruses as a mosaic of bacteriophage inherited capsid proteins. Why is that? Because as you know, adenoviruses belong to the PRD1 adenovirus lineage. These are viruses infecting all sorts of organisms throughout evolution that build their major capsid protein with a double jelly roll located perpendicular to the capsid surface. So it is thought that they all come from a common ancestor that was probably a bacteriophage, similar to detective viruses like PRD1. So the major core protein would have come inherited from a common ancestor. And now we see that adenoviruses have a decorating protein on the capsid that comes from a different kind of bacteriophage, the tail phages. And this, we think, was probably picked up during evolution by sharing the same habitat with bacteria of bacteriophages, for example, in the gut of the infected animal. So this was a low resolution cryo-EM study in combination with the crystal structure of recombinant protein. But to see LH3 in its biological context in the virion, we went ahead and determined the high resolution structure of the lizard adenovirus by cryo-EM. This is a 3.4 angstrom resolution structure. This is the icosahedral asymmetric unit as looking from outside or from inside the capsid. Inside, we have the inner minor code proteins and some unassigned elements that we attribute to the genus-specific proteins, P32K and LH2, but unfortunately, the quality of the density wasn't enough to unequivocally assign the identity of these elements. However, I want to keep focusing on LH3, the external decorating protein. So I showed you before the structure of the recombinant protein fitted on the 11 angstrom resolution cryo-EM map of the snake adenovirus. And here we were missing the 30 amino terminal residues in LH3, which were absent in the crystal structure. Now we have the high resolution structure of the whole virion, and we can see how the beta helix domain of LH3 connects to the capsid surface by these extended amino terminal domains that join together here on the capsid surface, forming this pedestal. If we look at the pedestal from the capsid surface, we can see that it forms a sort of Triskelion structure, and that it contacts the 3 hexon capsomers around it. If, for example, if you look at the pink subunit here, you can see that it contacts 1, 2, and 3 hexon capsomers. If we look now at the contacts at the beta helix domain, again, we see that the pink monomer contacts 1, 2, 3, and 4 different hexon monomers. In summary, each LH3 monomer interacts with 6 different hexon monomers in 3 different trimers. And we can see how this is probably contributing to the increased thermostability of the adenovirus capsids. So remember that I told you that LH3 might be substituting for the role of 9 in the architecture of the adenovirus capsid. And this is so because the pedestal region of LH3 occupies the same positions in the capsid as the Triskelion region of protein 9 in human adenoviruses. You can see here a comparison of the structure of both proteins. You can see that there's carboxy terminal domains are very different. One has the trimeric beta helix domain, and the other one, protein 9 in the human virus, wanders away, crawling on the capsid surface to end in this alpha helical cluster. But if we focus on the pedestal or Triskelion domain, we can see that they are very, very similar between the 2 viruses. We have this tyrosine triplet here and the hydrophobic residues down here, forming a hydrophobic core that stabilizes the Triskelion and it's binding to the capsid. If we look from the other direction, from the capsid surface, again, we can see that this Triskelion is very similar. And if we now look at the sequence after knowing the structure, we can indeed find the conservation of this hydrophobic patch and also this tryptophan here at the corners of the Triskelion. So let's now look at these proteins in the context of the mastadenovirus and adenovirus genome. It turns out that it was known for a long time that LH3 occupies the same region of the genome as a protein in human adenovirus called E1b55K, and they had a certain sequence similarity in their carboxy terminal domains. Actually, when the first adenovirus was sequenced, this gene was called E1b55K. 55K is not a structural protein in human adenoviruses. This is a protein that is synthesized in the early time post-infection to deregulate and control the cell cycle for the advantage of the virus, so that the virus, the cell, works for producing the viral proteins. Now we see that there is a similarity between the N-terminal domain of protein 9 and the N-terminal domain of LH3. But this N-terminal domain is upstream of the C-terminal domain of E1b55K, which is similar to the C-terminal domain of LH3. So what's going on here? Are these bits and pieces of the genome jumping around back and forth? What we thought was that maybe we were seeing here an ancient gene duplication, that maybe from the LH3 gene in adenoviruses, the gene had been duplicated, and then each copy would maybe have evolved separately to produce in the human adenoviruses the 2 copies, E1b55K and 9, which one had conserved the amino terminal domain and the other would have conserved the carboxy terminal domain. To figure out if this hypothesis was indeed correct, we contacted Mart Krupovich in Paris, and Mart did an extensive analysis and comparison of the adenovirus genomes, and also he compared these adenovirus genomes, this region of the adenovirus genomes, with other genomes in the databases, and he found that indeed the LH3 gene and the E1b55K are bona fide homologs. And also LH3 is also related to bacterial peptide lyase enzymes and bacteriophage tail spikes. Interestingly, this analysis also showed that there are some mammalian adenoviruses, mastadenoviruses infecting horses that still retain 2 copies of the LH3 gene, showing that indeed a gene duplication has taken place. In the horse adenoviruses, one of the copies has already lost the Triskelion region, and so this copy probably cannot any longer bind to the capsid. And finally, in the human adenoviruses, the second copy of LH3 has lost all the trimeric beta helix domain, turning into protein 9. And the E1b55 copy has acquired this long N-terminal extension that would also hinder its incorporation into the capsid. So in conclusion, we have shown that the adenovirus LH3 protein has a fold typical of bacteriophage tail spikes, making adenoviruses a mosaic of bacteriophage inherited capsid proteins. Extensive interactions between LH3 and hexons stabilize the adenovirus capsids. And with the high resolution cryo-EM structure of the lizard adenovirus, we have shown that the evolution of this LH3 gene to become 9 and E1b55K in human adenoviruses has gone through a process of first gene transfer to acquire the gene from bacteria or phages, then gene duplication, and finally an exaptation event where the E1b55 gene has turned from a capsid protein to a cell cycle regulating or deregulating protein, an oncogene in summary. And finally, these are the people involved in the work here in red, and also the collaborators working in this particular subject. Thanks for your attention.